So, distinguished guests, ladies and gentlemen, of course, as the rector of the University of Bonn, I'm very proud to co-host this evening together with Dorothee Zwonik, the Secretary General of the German Research Foundation. And the two of us, of course, and Ms. Zwonik will introduce later on our guest, the two of us, of course, we are very proud to have a prominent member, not only of the SAB of Ban Ki-moon here, Professor Hoch, but also a renowned scientist in the field of microbiology. And so this will be introduced later, but of course, since I'm a geneticist, so I'm also coming from the field of biology, so I've asked myself whether the global challenges that we're facing, whether those could be somehow addressed by microbiology. And so you may have seen this in the press recently. There were two important papers in a scientific journal, which is very prominent internationally. This is called Science. So one paper was about a paper of Craig Venter. You may remember him, with his Human Genome Project. And he has started a couple of years ago a new project where he has tried to synthesize organisms and in fact bacteria. And they have in 2010 they came up with the first, let's say, microbe that they tried to basically create synthetically in a context of synthetic biology. So that had 951 or so genes, which they transplanted in a bacteria where basically the genomes had been taken out, so that to try whether these synthetically synthesized genes would enable the organism to live, and it did. So in an event, basically, a couple of afterwards, from 2010 to today, they have tried to reduce the number of genes from 951 to the minimal number that a bacteria requires to live. And they came up with 473 genes. The problem is that this bacteria only lives in the laboratory, but it cannot survive outside. So although they have really tried hard to identify the essential genes, but in fact they have missed a couple of genes which in fact have redundant gene functions, and they don't know what these genes, the other genes, are doing, but they know that they are essential for a bacteria to live outside of the laboratory. Now, this is quite remarkable, right? But the second paper is even more remarkable, because that may have to do with these environmental challenges that we're facing, in particular, the plastics, the topic of plastics, plastic material, which is polluting our world, in particular the sea. There was a Japanese group from Kyoto who identified a bacteria that was able to digest plastic. This bacteria, which they isolated from a certain source, had two enzymes encoded by the genome that were able to cut plastic into pieces, if you want, to digest plastic, to use that as a carbon source for growth. And that is remarkable because we have to face that most of the bacterial world is still basically not really understood, because more than 90%, and Professor Hoch knows this very well, cannot be cultivated in the laboratory. 90% of the bacteria cannot be cultivated. So you know that these bacteria exist because you can isolate their DNA, their genome, but you cannot keep them in the lab to basically try to understand what their physiology is. And I think one of the big challenges for the field of microbiology is to make these bacteria cultivable, so that one could understand what kind of capabilities they have. And I think, so I expect that this rich source of bacteria will solve a lot of problems in this world if we could basically explore it or discover new features. And I think this is a challenge for this field of microbiology, a fascinating challenge. And so with this, so microbiology can be at least in part a solution to some of the problems. With this, I would like to hand over to Ms. Dorothee Zwonek, who will introduce our guest today a little bit more detail. Thank you.